I am nervous, I am proud, I am happy to welcome Julio Fernandez, who is a distinguished professor at Columbia University. He is also my postdoctoral advisor, who actually invited me to come to New York City in the first place. So this is sort of a nice 10-year later celebration of me being in New York. And we're not far in terms of geography, and you will notice some similarities between the research that he's going to talk about and the things that you guys hired me on. But I wanted to talk to you first about why Julio Fernandez is such a distinguished fellow. And the reason is that he's the pioneer, he's basically the founding father of single molecule force spectroscopy, and he's the first one to consider the profound question of can we look at protein folding at the single molecule level. When you do chemistry, when you do biochemistry, you tend to do experiments in beakers or buckets, and you tend to mix reagents and look at the reactants and look at the products, and then you try to infer what the mechanism of those chemical reactions are, but just by looking at their bulk biochemistry behavior. Now, what Julio did to revolutionize and sort of transform the field is to say, what if I catch these molecules in the act of doing their reactions? So he is looking at single molecule chemistry and protein folding reactions, one molecule at a time. You have to sort of imagine in your head that these are nanometer objects and that you need very exquisite control and precise experiments to be able to measure these kinds of phenomena. And so one of the things that has always amazed me about working in Julio's lab is the rigor and the sort of statistical nature and accuracy of the kinds of experiments that we were doing. And the other thing that's amazing is that he has, throughout his career, remained a sort of pioneer. Every time he publishes a paper, it transforms the field in some way. So he starts to take paleoenzymology to the single molecule level, or he looks at how can we enforce protein folding so that we can make stronger muscles or weaker muscles. How can we do molecular design to actually have control over the mechanical strength of molecules. So you see here yoga on the picture here. So I just wanted to say that on a more personal stage, Julio has been very influential in my career. I can safely say that I would not be here today had I not done a postdoc in Julio's lab. One of the ways in which that happened was that he placed a guillotine above my desk and said, you're going to make great things happen. And so that has really revolutionized the way that I have been doing my science. So I have a great debt to Julio because he has been very important in my career path. But more than that, or more broadly speaking, every single person who was a postdoc in his group during the time that I was there is now I wouldn't say famous, but is a professor of something or other, trying to get to that same curiosity and contagious scientific research level that Julio kind of diseased us with, or how should I put it? We all got the bug, and now we struggle with it. So that's one of the things that I certainly feel indebted to Julio for, and I guess I'm taking up a lot of time, but I just wanted to say all these things. Last but not least, all these people, Frauke, Sergi, Raul, myself, we're all professors, but we're all also deep friends. And I think that that's something that says a lot about the environment that Julio creates. So thank you, Julio, and let's hear the new science.
Thank you, Jasna. It's a pleasure to be here. And
you have 5 minutes left.
Yes. Exactly.
I want to tell you about protein folding and yoga. And that's kind of what I'm going to try and explain today. This is how science really impacts our understanding of living organisms, and that really is my motivation. You can see here one of the asanas which is all about stretching muscle. And we have really no understanding what stretching does. And we know it's good, everybody does it, we're not exactly sure why we do it, and I hope to give you some idea of perhaps what is really happening. One of the amazing things is to watch a baseball pitcher throw a ball that reaches 105, 107 miles an hour. It's hard to imagine a faster human motion. The rotation of the humerus is the humerus bone. It's the fastest motion that you can do with your limbs. And but you cannot sit on a chair and throw the ball like this. You have to have this sort of orchestrated dance where you move your entire body and you store mechanical energy in a distributed set of muscles. And then you harness very rapidly all of that mechanical energy into one very quick shot, allowing the ball to exit at a very high velocity. And you say, I'm a baseball enthusiast. I have to tell you, I hate baseball. However, I'm profoundly amazed at how these people manage to do it. And here is just a movie of a professional baseball pitcher. Look at the entire, this is 1,000 frames per second. He's storing mechanical energy in the motion. All of this is going into muscles in the body, which are being stretched. And then that final humeral rotation is when the ball is launched. There, now. Without the stretching, this doesn't happen. So, how does this work? Where is the mechanical energy being stored, and how, and why? Right? We've been doing this for tens of thousands of years. The hunting, throwing an atlatl, right? I mean, it requires the exact same motion. Throwing a stone, I mean, I'm sure you've all reached down to a stone when you have a dog that's looking kind of iffy, right? The dog knows. They run away right away. We've been doing this for a very long time, right? Throwing has been absolutely essential for us. But is that trivial? I mean, do you really know what you're doing when you're stretching and you're throwing, when you're contracting a muscle? It's surprising how little we understand. We all know that there are myosin motors that contract and generate force. We know that there is actin, but we have very little understanding of how the mechanical energy is stored, retrieved, and used this way. So, the protein that's responsible for the storage of mechanical energy is called titin. It's the largest protein in the human body. It's up to 36,000 amino acids long. It's a micron in length. It straddles the sarcomere from the C line to the M line, and is the third filament in muscle. The titin gene, it's 100 kilobases long. It codes for proteins that are alternatively spliced, so that you have over 500 different types of muscle in your body. Each muscle has a unique alternative splice form of titin, meaning that there are different exons in the gene that are combined to create titin isoforms that have different elasticity. And so it's as if evolution has very finely tuned the elasticity of each one of the muscles in your body. Right? How much stretching you can gain from this, how much energy you can store in each one of the titins. It's all tightly regulated. And one thing that's interesting to consider, when bears hibernate, their heart rate drops to less than 20 heartbeats per minute. And together with that, they change the elasticity of the heart muscle wall so that the heart doesn't dilate too much. And they do this by switching isoforms of titin from a soft isoform to a very stiff isoform, which is done just at the moment of hibernating. And then at the end of hibernation, it switches back so that the bear can actually have normal elasticity. I think training, I think yoga, I think athletic performance, it's all related to this protein, which over the next 10, 20 years probably will inform us of much of how muscle works in a human body. So you take apart a muscle, it's made of these fibrils. If you look at the fibrils with a microscope, you see the striations. The striations, when you look at them with an electron microscope, and this is about 3.5, 4 micrometers in length right here. The striations are made of these bands, and this region here is where you have the thick filaments of myosin, and this region here is called the I band. That's the elastic region of the sarcomere. This is a Z line, this is another Z line, this is the M line right in the middle, and a single titin molecule will reach from the Z line to the M line and would be the origin of the elastic part of the sarcomere, and would also be the template organizing the thick filament where the myosin heads are. So you're probably used to seeing these schemes in textbooks. They generally don't have titin. It takes a long time for textbooks to catch up on things. Although titin's been around for 20 years and is the largest protein in the human body, it had not been seen because biochemists typically run gels where they can see the proteins, and titin was so big that it always remained at the top of the gels. It's only when Quan Wang ran a very low density gel that he actually got this protein and identified it. And that was a little over 20 years ago. So now we know that titin is the third filament, and this is more or less the structure, the molecular structure of a single contractile unit. You have the actin filaments, which don't change and are always more or less the same length, a little over a micron. You have the thick filament, which have over 250 myosin heads poking out in a helical way, reaching out to a hexamer of actin filaments. And then you have the titins. This is only in 2 dimensions, so you should be seeing that there are 6 titin per thick filament, and this is a hexagon around the thick filament that then slides in and out of this panel of actin molecules. Muscle is a remarkable tissue. If you look at the stretching, this is a relaxed single myofibril. You can stretch it and relax it and stretch it and relax it, and the structure never changes. It never comes apart. It retains its three-dimensional architecture, and you can see that in this case we're increasing the amount of force on titin molecules, and we're decreasing it, and the muscle regains its shape and its length. And so muscle is elastic tissue, and the elasticity of the muscle cells is caused by this protein titin. So
when you warm it, does it get shorter or does it get longer?
The temperature range here doesn't change very much. The temperature range in a muscle is pretty narrowly defined, and so this is not going to change very much in the cell. If you do it in vitro outside, it doesn't really change very much. I mean, the polymer elasticity, which is the source of much of the recoil, is referred to absolute temperature. So you have to change the temperature very, very much to see any effect. So titin is made of hundreds of these tandem modules in series, and when you apply force, these modules unfold and refold. And over almost 20 years of research on force spectroscopy, we've learned a few things. One is that you can pull on a protein. This is a tailin molecule here, some work that I did with Mike Sheets a few years ago. You apply force, you unravel the tertiary structure of the protein, and then one thing that you do is that you expose some binding sites or some reactive sites that are normally hidden inside the protein. But when you pull on the protein and you unfold, these sites become exposed and they can actually interact with molecules in the environment. You also have these folding reactions, so that if you reduce the amount of force here, the protein is going to go back to its original shape. This is totally reversible reaction, and I was talking to some of the graduate students at lunch, after lunch, and this happens in the absence of chaperones or any other assistance. It's just completely spontaneous. You You also have some chemistry that's pretty interesting. Typically, you have thiols. Thiols might form disulfide bonds or might react with molecules on the outside, such as glutathione. And when you have this chemistry, which is cryptic chemistry, the thiols have to be exposed by unfolding. You also have effects of these modifications on the folded protein. So you have a fine interplay between folding and unfolding, elasticity, and chemistry, all of which contributes to create what I call a mechanical battery of sorts. You store mechanical energy in titin that then you can use, for example, the pitcher for throwing the ball. So how do we study this? And these are sort of standard techniques. By now, we create titin-like molecules in the laboratory. This is my laboratory at Columbia, and where we make tandem repeats of modules from titin, and then we use chemical anchors to attach the protein, the titin-like proteins, to glass and also to beads or to an AFM tip. Nowadays, we're using this technique called halotag, which was developed by Promega. It's an enzyme that reacts with a chloroalkane ligand, forming an intermediate which is covalent. So you can very easily attach molecules now to glass surfaces using this chloroalkane halotag technique, which works extremely well and that we use now as standard in our laboratory. One instrument that we started with and we're still using is the FM. We made modifications of the FM so that we could nowadays, like I explained, anchor our proteins to a glass surface with chloroalkane, so we get a covalent anchor here, and we use thiol chemistry to attach the other end of the protein to cantilevers, and that way we can stretch molecules and make measurements on the relationship between force and length for individual titin molecules. And so from force and length we can calculate work and measure energy and create energy landscapes and make predictions for the elasticity and the storage. So the FM is remarkably simple. Most of you probably know how it works. You have cantilevers, you have a laser beam that bounces off the tip of the FM. We have a high-resolution piezoelectric actuator that moves the protein away from the cantilever, and that can give us a measure of force and length.
Sorry, what is the protein in that figure?
Right, so the protein would be on the surface of the piezoelectric actuator, and we would press that surface against the cantilever, and then the end with the thiol of the protein would attach to a gold-coated tip of the cantilever. And that's how we get the anchor. The other end is anchored nowadays using the halotag. So you can do these experiments of folding and unfolding. Here is a typical experiment with a titin-like protein. We would just apply force, and you get step increases in length. Each step marks the unfolding of one of the modules. We typically make proteins which are 8 repeats because that's what bacteria can easily produce. And then you quench the force down. You allow some time for the protein to refold. You pull again, and you see that the protein has recovered its mechanical stability. If you pull too soon, fewer proteins have recovered their mechanical stability, and if you pull very short times, none recover their mechanical stability, and you just get the elastic extension of the polypeptide. You can do this for fairly long periods of time with the FM, up to 10-15 minutes in length, roughly. But then the mechanical drift becomes too significant and you have to stop and recalibrate. So that's more or less the time scale. But we learned a tremendous amount using this technique, and we learned that mechanically we could unfold titin, and we could refold titin, unfold titin, refold titin ad nauseum, and that this will be a very significant part of the mechanical behavior of this protein. My cell biology friends, of course, would always complain that they think that there's no unfolding of proteins in vivo, that unfolding is bad, it's toxic, it's something that's undesirable, and so that we had no proof that titin actually worked this way, and that perhaps unfolding just never took place, and that these happen only under extremely high forces. And so to address these issues, we studied the behavior of titin in an intact muscle. And the way we did that is that here you have a cutout of a sarcomere. Here is the thick filament. I'm only showing the elastic region where you can see the titin molecules. And so my collaborator from Germany, Wolfgang Linke, has made some antibodies that recognize specifically some regions of titin. In particular, the 39th module of titin, which is universally expressed in tissues of the rabbit. This is a rabbit skeletal muscle, a rabbit psoas muscle. And so when we label a myofibril with this antibody, the antibody is attached to a quantum dot. So what we see is the fluorescence of the quantum dot, which gives us a tremendous amount of light. And so we get two bands of light around the seed disk. And you can see here the two bands, more or less. This is a fairly high density of staining. But if you decrease the staining and decrease the staining to the point where you just get a few quantum dots attached to a muscle fiber, then in a few cases we're able to get quantum dots, single quantum dots across the C line. You can see here in regular microscopy we can identify where the Z line is from an extended myofibril, and then when we switch to fluorescence you can see the two quantum dots attached to I39 on both sides of the C line. And then if we track as a function of time the position of these quantum dots using centroid fitting, and we do all the programming in the laboratory, then you get movies like this where you see how much time the quantum dot spends at any particular position. And so this is the separation between the two quantum dots. And you can see that, actually, if you look at this recording sideways, you can see that the sampling reveals already that the position is not continuously sampled, but rather that there are discrete states in the position of the quantum dots. And these states are separated by between 11 and 20 nanometers. Yes?
How are you optically resolving 20 nanometers per pair?
We don't. So what you do is that you do centroid fitting. This is a technique called Fiona, where basically the single quantum dot will give you sort of a Gaussian, two-dimensional Gaussian distribution. You fit a Gaussian distribution to that, and you track the center of the distribution. This has been shown to work extremely well for motors.
But you have two overlapping Gaussians, and you can resolve them?
Right. We fit the two Gaussians with a multi-peak function, and we can. The distance is around 500 nanometers or so it's big enough to give us well-separated points.
Sorry.
Yeah, you see the distance.
Separation distance. Sorry,
Yeah.
yes, okay, I'm sorry.
Right. So they're not 11 nanometers apart. They change in, end-to-end distance by 11 nanometers, between 11 and 20 nanometers. So we get these histograms, and we've done that in a few myofibrils, and we observe a similar distribution of sort of discrete states from these quantum dots. We make a histogram with all our data, and we see that the length of titin in this intact tissue change in steps of around 11 and 22 nanometers. And so we wanted to find out if this had any relation to the observations of titin folding and folding that we had made with the FM. However, there is a problem. The forces in the FM tend to be pretty high, because when you operate with the FM, you have good control of the forces. You're pulling at forces of, let's say, 100 piconewtons or 200 piconewtons. But the forces that we calculated for an intact muscle were in the range of 5 to 10 piconewtons, which means that everything happens on a much slower time scale. But we didn't really know how to scale down the data from 100 piconewtons down to 10 piconewtons. So in order to do this, we developed, in collaboration with some colleagues from the National University of Singapore, a technique where we combine magnetic tweezers together with the halotag anchor chemistries that we had developed. And this is the work of Janelle Popa and Andres Rivas in my laboratory. And the magnetic tweezers are extremely simple. Basically, you just bring a pair of magnets, N52s, ceramic magnets, which are very, very strong. And then you put them right on top of a paramagnetic bead, and you can generate forces up to a couple hundred piconewtons this way. I mean, it's very easy. The beads are about 3 micrometers in diameter. And then you tether the polyprotein, the titin-like protein, with a halo tag to glass, like before, and then we have a biotin streptavidin anchor to the bead, which actually is remarkably robust, and it works very well. So we can do this, and here you see some of the chemistry that we do. We silanize the glass, use chloraldehyde to link to the chloroalkane ligand, and then the chloroalkane ligand binds covalently to the halo tag protein, anchoring firmly our protein to the glass, and then we flush in some paramagnetic beads, and then the beads that lift can be pulled and observed as a function of time with a CCD camera that measures the distance between a reference bead on the glass surface and the magnetic bead. And doing that, we can track beads with nanometer resolution for day-long experiments. So you grab a single protein, you can track the single molecule for an extremely long period of time. Very stable. The instrument is very simple. Here you see the camera. You see the magnet positioner here. And most of it really is programming. The magnet position is determined by a coil system that's like a loudspeaker, and that moves the magnet up and down following a pulse pattern that we encode in our computer. So it's really very easy, and you have a magnet law that tells you exactly the relationship between magnet position and force, and so at all times you can apply a known force of arbitrary shape to your molecule. Most of the effort really is in the programming. Here you see the reference bead and the paramagnetic bead. Here they have different colors, so we can tell them apart. And then we take the Fourier transform of the beads. The Fourier transform is really useful because then it's not sensitive to the position of the bead in the field of view. And then we get a radial vector out of the Fourier transform, and by correlating the radial vector of the current image with a radial vector stack, say, prior to the experiment, we can measure the actual position of the bead with nanometer resolution and with drift correction. So we can do these experiments that can last a long time, where we can apply force, unfold the protein, relax, let it fold, unfold again. But in contrast to the 15 minutes of the FM, we can do these experiments for, like I said before, for up to a day-long set of recordings. That creates a problem because if you have a molecule that's working really well, that single molecule ties up the setup for an entire day. So the scale has changed in the laboratory. So here are some experiments that tell us what happens then when we look at titin in the force range where muscle actually contracts. You can see here, this is one of the domains that unfolds when you stretch titin, as I showed earlier. And you can see that this is a tandem repeat of 8 identical repeats of the 10th module of titin. And we apply 54 piconewtons. And you see 8 steps of unfolding here. And then we relax the force down to 4 piconewtons. And that triggers an immediate collapse of the polypeptide and then folding. The force is too low to see any detail here, but the protein folds, and then we unfold again, but this time at 6 piconewtons, and we reach some sort of an equilibrium where you can see the fluctuations in the length of the protein, which corresponds to transitions between collapse, molten globule, and native states. And I'm going to explain the details of this in a moment. But each one of the modules in titin has a completely different mechanical stability. So if you take another module, I91, which is in a different part of the elastic band, and you do the same experiment, it takes a higher force to unfold more or less at the same rate, and then you quench the force and you apply the same 6 piconewtons as before, but now it takes about 3 hours to see the first unfolding event. So, they respond to force very differently, and you have hundreds of these modules that provide sort of a graded response to force that make them, the recruitment of these modules at any particular force will vary in time greatly from one module to another. More interestingly, though, is this experiment here, where instead of quenching the force very low after unfolding, we quench the force to a relatively high value, 6 piconewtons, which is right in the middle of the range where the muscle works. And now you see an immediate collapse, which is the recoil of the entropic elasticity of the polypeptide, relaxing from the high force to the 6 piconewtons, much lower force. But now you see, after the collapse, you see a series of steps down, which correspond to transitions from the collapse to the molten globule state of the protein. These are actually getting shorter at a force of 6 piconewtons. So notice that the protein is actually doing mechanical work on the probe as it folds. And if you count the number of steps down, and then you pull again at a high force, this number of steps down is always equal to the number of steps of unfolding. So very clearly, these steps down correspond to the folding of the titin protein at 6 piconewtons of force. And you can do the exact same experiment with I-91. You unfold I-91 at a much higher force, but then you quench the force to 5 piconewtons, and you can see the steps here of the contraction of the protein as it goes from the collapse state to the molten globule state, doing mechanical work on the system. And you can see again that in all cases, the number of steps down matches the number of steps up on a subsequent pulse. So very clearly, this is related to the folding of the protein. One thing that you might notice is that these steps are smaller than the unfolding steps. So the unfolding steps here are about twice the size of the folding steps. And that's just because the force is very different. If you measure these unfolding and refolding steps at various forces, you can plot the step size as a function of the pulling force in piconewtons or magnet position. And you see that you can describe this very well using the worm-like chain model of polymer elasticity. That means that if you unfold at a high force, the step size is going to be, let's say, 20 nanometers, because the polymer is going to extend much further when it unfolds. And then you quench the force, and then the folding step is going to be scaled to a smaller size because the force at which you're working is much lower than the force that you use to unfold the protein. So it scales very nicely with a simple theory of polymer elasticity. And so that's how we explain the experiments in the intact tissue. If you unfold a protein now that's a tandem region taken out straight from the native titin, and so this is a protein that encompasses domains 4, 5, etc. up to 11, and do these experiments, at 6 piconewtons of force, you get a step size distribution that matches pretty much what we saw in the intact tissue. So it means that the steps that we saw using the quantum dots in the intact muscle are originating from folding and folding reactions in the titin molecule that's taking place at 6 piconewtons for physiological extensions of a muscle fiber. That means that unfolding and refolding occurs in the intact tissue under physiological conditions all the time. What does that mean? What are the implications? If you take then at face value the experiments and say, all right, so I admit that folding does occur together with unfolding in the intact muscle tissue, how is that translating to anything of interest for the pitcher that's throwing the ball? Well, so if you look at the folding dynamics of a titin protein, you can see here the unfolding steps, right? And please notice that the time scales have been clipped here so that you don't have to wait forever. So this is on a logarithmic scale here to see all the steps of unfolding which occur exponentially. And then you can see here, when we quench the force down to 5 piconewtons, you get the instantaneous elastic recoil, you get then the steps down of the collapse and folding transition of the protein. And then, if you leave the force in there, you reach some kind of a steady state that can go on for hours. And so, I'm just going to focus on this folding contraction. So, what is the probability of this folding contraction as a function of force? It's very, very narrow. So, you can do the experiment of collapsing and folding at various forces below 10 piconewtons, and you see that it's very sensitive to the force. You don't get the folding contraction. In all cases, you get the elastic recoil, but you don't get the folding contraction until you fall below 10 piconewtons. And then very rapidly, between 10 piconewtons and 5 piconewtons, it's all over. So, I mean, it falls very, very fast as a function of force. And if you plot the folding probability then as a function of force, you see here that it goes from 0 to 1 between 8 and 4 piconewtons. And this is pretty much the same for all the modules that we have studied here. So although the unfolding can be extremely different depending on the type of module, the folding reaction seems to be generic and seems to be similar for all the modules. I show you that I-91, if we try and unfold it, it takes 3 hours to get a single module unfolded at 6 piconewtons. Yet it falls over the exact same range that the I-10 module, which readily unfolds at 6 piconewtons. So the recruitment of the proteins to the unfolded state can vary tremendously, but the folding reaction seems to be the same for all. And it's following this red line here. So it's just a beautiful sigmoid between 8 and 4 piconewtons. What does that mean? Well, if you're at 8 piconewtons and you fold in a single step, that single step contracts the length of the protein by 10 or 12 nanometers, depending on the force you're at, right? And that means that the folding is actually doing mechanical work, because you're pulling at 8 piconewtons, yet the protein is getting shorter in a single step. And that amounts to a surprisingly large energy. A septojoule is 1 piconewton per 1 nanometer. And so we have over 100 septojoules of mechanical energy delivered by a folding protein at 8 piconewtons. And that's much, much larger than the energy generated by a single myosin motor that generates 38 septojoules under the same conditions. So this is rather surprising, and it's never been considered before, that protein folding can actually do mechanical work far in excess of the work of the myosin motors, which so far have been thought to be the only source of contractile energy in cells. And so, but that's not good enough, because the probability of observing steps at 8 piconewtons is pretty low. You see that we're practically at the end of the folding probability curve here, right? So a better way of understanding this is to calculate the expected value of the contractile energy. And so for that, we multiply the folding probability times the step energy delivered by folding. And that's this curve. The black curve here is the expected value of the contractile energy. That means that if you have a huge number of molecules, on average, how much energy you expect from folding at what force. And you see that it peaks at 6 piconewtons, which is quite remarkable, and the peak at 6 piconewtons is already higher than the energy delivered by a single myosin motor.
Excuse me, when you say folding probability, it's over a given time? How do you define folding?
60 seconds.
60 seconds.
Right. So if we did the experiment over a longer time scale, it'll shift a little bit, but not by much, because we're getting almost 100% folding. So it'll probably shift maybe half a piconewton lower if we waited much longer.
Is stored energy before you can release it, you have to store it.
Of course, you have to stretch.
When you store it, it is done by motors. division?
Well, you have all kinds of things contributing. Gravity is one source. We move in a gravitational field. So by doing motion, doing work on the gravitational field, some of that energy can be stored in your muscles. And so anything that stretches titin will store mechanical energy that then you can collect back during the motion. Remember that when you contract one muscle, you're extending another, always. Okay? And so when you contract one muscle, the other one is simply passively stretching your titin molecules, and then you get back some of that energy when you move it the other way.
But when I contract, I still have to perform work by motor.
Let me explain how it works, okay? Think of what I just told you. I just showed you data that the folding, if you recruit a bunch of these modules to be in the unfolded state, now we ask, how much energy do we get back when we fold? So here is a summary of the data that I showed you. The red curve reflects the folding probability as a function of force. So if you're at 10 piconewtons, very likely you're going to stay unfolded. Nothing's going to fold. But if you go down from 10 piconewtons to 4 piconewtons, everything folds. And it's pretty generic. So anything that's unfolded will fold at 4 piconewtons, right? Yes?
I'm just curious about how this gets unfolded in the first place, because it looks like at 10 piconewtons it stays, if it's folded, it stays folded. Right?
If you unfold at 10 piconewtons, okay, so think of that baseball
Right, but it looks like it
pitcher,
takes 100
right?
seconds.
That baseball pitcher is distributing, mechanical stretching over a number of muscles. That stretching area is consuming energy from his entire body, right? But he's harnessing that back in one particular motion to throw the ball. So how do you get unfolded? You get unfolded maybe slowly with that kind of motion of the body, and then you recover part of that energy in the pitch.
I guess I'm just trying to reconciling that description with the previous graph, which shows like 100 seconds unfold at 60 piconewtons, right? It seems like you're not putting 60 piconewtons.
Well, you have hundreds of modules, right? And so in this particular case, we're working with a module that has these kinetics.
Okay.
There are many other modules that unfold much faster,
So the
and
answer is
some other modules that unfold much more slowly, right? So if you have, let's say, in your soleus or your gastrocnemius muscle, in your leg, you have 150 Ig domains, right? If you grab the entire protein, which we have not yet been able to do, right? This is going to be way faster.
Okay, so that's
all right. It's just that I picked two examples and show in these time scales how they work, and I'm projecting from that to the entire muscle. I imagine that because you have 150, the probability of many of them unfolding is way higher than for only 8, right? Okay, so this is sort of the picture that I have. Think of a very simple model for how muscle contracts. Here you have actin. I drew a bunch of active binding sites here for myosin. You can see the yellow things, right? And there's a little, cucumber-like thing that fits right in there, right? This is the myosin motor. And the myosin motor, when it cycles ATP, it does something called the power stroke. Basically, it binds to actin and then it shifts. And it generates force. And people have made measurements using optical tweezers of the force generated by these motors. And they take steps of around 10 nanometers and generate forces of around 4 piconewtons. That's kind of what comes out of 20 years of experimentation with single myosin motors using optical tweezers. That is remarkable because now we know that there's titin in there. And it's inescapable that these molecules work together. So think of the consequences of what I just showed you. If you're just hanging there and your muscle's not contracting, let's say that you're applying 10 piconewtons of force, which is a fairly reasonable number for any kind of muscle activity. You can calculate that by simply dividing the stretching force by the number of titin molecules and the cross-section of the muscle, which is all very well known. So 10 piconewtons is a fairly standard number. If your muscle is being stretched by 10 piconewtons per titin molecule and the motors are not engaged, 10 piconewtons will cause unfolding, right? Now, when the motor engage here, and you're all physicists, you'll appreciate that now the force on the titin molecule is decreased by the amount of force generated by the motor. So if the motor generates 4 piconewtons, the force on titin drops from 10 piconewtons to 6. And at 6 piconewtons, you get substantial folding, right? So, in a sense, the only role of the motor here is to straddle the folding probability of titin in such a way that when you reduce the folding force, the unfolding force, from 10 to 6 piconewtons, now you're going to go from here to here, and folding would actually deliver mechanical energy that aids the force of contraction of the muscle. In a sense, what I'm trying to say is that you cannot extricate myosin from titin. These are two molecules that work together, both in the unfolding, storing mechanical energy, and then in the contraction, delivering mechanical energy. In this scheme, I just show you the actual energy delivered by folding is bigger than that delivered by the motor. And you can see how the scheme could actually work, right? I mean, it's pretty obvious that the motor here would just reduce the force on the unfolded titin, straddling the folding probability and delivering a mechanical contraction. What's remarkable is that people for 2 decades have been measuring the relationship between force and sarcopenial length. Lots of people have measured this for every different type of muscle in the body, in animals and even in humans. This is data from humans. The soleus is right here on your legs, heart muscle tissue, vastus lateralis, it's a muscle that runs through here, is about 2 inches square. And so the stiffest tissue is heart muscle. And so you can see that the sarcomere length needs to change only by about 100 nanometers. It's nothing. Okay, so the entire sarcomere is maybe 3-4 microns, but only a change in 100 nanometers in sarcomere length shifts the force from 4 to 8 piconewtons. You can see here that very little change in sarcomere length changes the force from 4 to 8 piconewtons. The softest muscle, soleus in this case, you need to shift the force quite a bit. So we use this data, and this is not the original data, these are fits to the data from the literature. We use these functions to create a reconstruction of the folding probability versus sarcomere length for the intact muscle. And you can see here, these are the 3 curves of the folding probability that I had plotted here as a function of force, but now I'm plotting it as a function of sarcomere length. And so the blue is for soleus, the yellow is for vastus lateralis, and the red is for heart muscle. What is remarkable is that people have also measured the active force generated by a muscle at different sarcomere lengths, and that's the black line here. This is actually a universal curve. Every muscle works the exact same way. You take any muscle and measure force generated by tetanus as a function of sarcomere length, they all superimpose pretty much, and that's this black curve here. What is remarkable is that the folding probability here falls exactly around the region where the peak force is generated by a contracting muscle. We know that the heart muscle operates in the ascending limb of this curve. We know that the longer soleus gastrocnemius muscles operate on the descending limb of this curve. So it appears that the folding probability curve for titin modules takes place for each muscle exactly at the optimal operating point. It's hard to imagine that this is just an accident, and it's very likely that then protein folding contributes with a very significant component to the amount of force generated by a contracting muscle, right? In that sense, recruitment of unfolded titin domains, it's crucial for maximally taking advantage of the amount of energy generated by the myosin motors, because all the myosin motors are doing in this scheme is relieving the force on titin and allowing titin to fold, generating a very important component of the force. Of course, I'm going to get into big trouble with this because, A, no one's ever said this before. B, the muscle field, like all fields, are totally encrusted with the notion that all you need is actin and myosin and nothing else. And C, the paper's been reviewed. I'll find out. I'll tell you next time I come back here. Probably with a black eye and some, a missing ear, and that kind of thing. But let me just put it this way. It's absolutely inevitable, in my mind, that protein folding generates a very substantial, if not the majority, of the contractile force of a muscle. I think that's interesting. How are we doing with time? 4:55. I'm just kind of warming up. All right, let me go to yoga then. So what is stretching doing? Stretching is recruiting a pool of unfolded modules that you can use to generate force during a contraction. You're totally stiff. Imagine that your titin is completely folded. Then the force of a contracting muscle is solely the force generated by the motors. And you get about half of the total. That'll be the 4 piconewtons that I showed you for a single molecule, right? So that'll be only 38 septojoules. But if you have folding together with the motor, you triple the amount of mechanical energy that you can then deliver by contracting muscle. I'm aware of the training, so no alarm here. So you want performance. If you want to optimize the way you distribute power between different muscles in your body, evidently it's to your advantage to have this pool, this reserve of unfolded modules, and I think that's what stretching does.
So you're saying you should be stretching before exercising rather than after?
I think that the pain that you experience exercising, if you have not done any stretching, is because the force per molecule is too high. And I think that stretching would allow you to extend the muscle passively without reaching a force that's too high. So that's one thing, one aspect of stretching that's important. The second aspect is that the more modules that you have unfolded, the more you can use this mechanism to increase the amount of energy that you deliver during the contraction. So I believe that is true. But folding is fairly quick. And so how do you want to modulate this? There are all kinds of novel chemistry which have come out of the work of my laboratory that also tell us a little bit how this might work. One thing that's very striking is the number of cysteines which are present in the immunoglobulin domains of the elastic region of muscle. There are lots of them. And if you look where they are, they tend to be buried in the structure. Some of them form disulfide bonds. Some of them are just buried in the structure. So these are the questions that I want to address in the next few minutes. Thiol chemistry has a big effect on protein folding, and as such, thiol chemistry then is very important on how you deliver mechanical energy in your body. The first thing is that we can do these experiments where if you have a disulfide bond in a protein and you apply force to the protein can only extend right up to the disulfide bond, and then you need a second chemical reaction to break the disulfide bond and allow the protein to extend. This is a movie made by one of my former students, Palab Kosuri, who's at Harvard now, and he will show that an enzyme comes in, a reductase breaks the disulfide bond. You get two steps. The first step is the unfolding and extension of the protein right up to the disulfide bond, but that cannot be broken mechanically. It has to be a chemical reaction. But if you have an enzyme, a reductase then can come in, and then break the disulfide bond, and you get a second step. So it's a fantastically simple assay for thiol chemistry. And so here you see an experiment with an enzyme done by Pallav. You can see that you have two kinds of steps, and if you make a histogram, you get steps of 11 nanometers and 14 nanometers. The 11 nanometer steps are from the unfolding right after the disulfide bond. The 14 nanometer steps are when the disulfide bond is exposed, the enzyme comes in, breaks it open, and then you get the final extension of the protein. So we have a very nice fingerprint for how the reaction proceeds. Now you allow the protein to fold and you pull again. We only get steps of 25 nanometers because we were using an enzyme that can only reduce disulfide bonds but cannot oxidize them. And so this is a reductase only, and you can see that on the second pull the protein folds correctly, but there are no disulfide bonds holding the length. But if you repeat the experiment with a right oxidase, this is PDI, which is an oxidase that exists in muscle, then you can see that now you unfold the protein, you get steps of 11 and 14 nanometers, you quench the force, allow the protein to fold, pull again, and you get now steps of 11, steps of 14. Those have successfully reoxidized the disulfide bond, but also you get steps of 25 nanometers, which are failures where the disulfide bond did not reform. So what I'm showing you is that there are these really sort of beautifully simple techniques to monitor the formation and reduction of disulfide bonds in titin modules. This happens for sure in vivo, and it's a very important way of controlling the free energy and the elastic energy of the protein. I did not show you how to construct the PMF of this, so I'm going to skip this slide. Let me just end with this final observation. So the introduction of a disulfide bond can greatly change the dynamics of folding and the amount of elastic energy that you can recover from the extension by limiting the extension to a length that's controlled by the position of the disulfide bond. Another way of doing this, which is what we call mechanical memory, and this is a work more recently of a former postdoc of mine that's now in Madrid, Jorge Alegre Cebollada, he found that even in the absence of disulfide bonds, if you have cysteines which don't react with each other forming a covalent bond, but that are exposed by unfolding, when these cysteines are exposed to glutathione, that is oxidized glutathione, glutathione is a tripeptide, it's a fairly large molecule. And typically in your body you have reduced and oxidized glutathione. That's the redox control system of cells. And when you have oxidative stress, let's say that you get bitten by a dog or you have a heart attack or some event that triggers stress in a cell, the ratio of reduced to oxidized glutathione changes. And you're going to have a lot more of the oxidized form of glutathione. So when you have oxidized glutathione, it can react with thiols, and it puts this sort of wart in the unfolded protein. And the protein cannot fold back. It blocks folding. So it can create the form of mechanical memory that can linger for a fairly long time. So you can see here, if you expose the protein to a solution containing oxidized glutathione for only 2 seconds, it folds readily. But if you expose the unfolded protein for 40 seconds in this case, and you fold for the same amount of time, nothing folded back. So you can completely block folding by having this chemical modification of an exposed tile. So that's mechanical memory. I mean, if you stretch and you unfold tight, and then you have some sort of reactive oxygen species modifying the tiles, that's going to alter the probability of folding, it's going to shift that curve, and it's going to change the elasticity of your tissue in a way that may be beneficial or detrimental depending on what actually triggered that oxidation. And again, I'm going to shift all of this. And we see that in intact tissue. I'm sorry, I'm going to have to skip here through all the way. And here by saying that... so when you hear elasticity, most of your physicists, you think of a rubber band. Titin, although it's responsible for the elasticity of muscle, needs to be thought of as a mechanical computer that has an algorithm to compute the optimum for the storage of elastic energy for each type of motion. And all our adaptation to running, to lifting, to moving in the gravitational field is related to the activity of this protein in a way that optimizes our energy consumption. Every motion is optimized so that we don't waste energy, because everything in biology is about being sparing with the amount of energy that you spend doing everything. If you don't have enough energy input, you die. So, optimizing the energy levels of consumption is very, very important. And certainly most of our consumption is related to muscle work. Titin plays a key role in this that is very poorly understood, and that you can imagine at a very intuitive level, if you're standing in attention for a very long time, you want titin to be folded. That way you don't need to be correcting your position all the time. You want to be like a tree.
Just fall over.
Just fall over, right? If you're going to run, this situation doesn't help you, and you want to stretch. If you want to do yoga, clearly it has an effect that's going to linger. I mean, you're not going to refold immediately your titin molecules. You want them to have some sort of mechanical memory that can be utilized for the motion that follows. So all of these things are related to this giant protein. And the physics of this giant protein determines the actual interplay in the energy consumption by the motors and what kind of mechanical work you do. My dream is to create a detailed model of titin that can be used to predict phenotypes, predict optimal conditions for physical activity, that can be used for not only for disease, but I think a far more interesting perspective is to think, how can we optimize human performance and human athletic performance in particular? I mean, there are mutations that make you sick. For example, point mutations in titin can give you dilated cardiomyopathies and you die. Maybe there are point mutations in titin that make you a better athlete, just because you can, store energy more efficiently, and you get a phenotype. It's a good phenotype. It's not a bad phenotype. It won't kill you. It will make you run fast. Right? This is entirely possible, and with massive genome sequencing and with these kinds of techniques, maybe we can begin to understand and clean up these possibilities. Thank you very much for your attention. Only in vitro, only in our assay. There's an enzyme called Glutathionylation, whose sole purpose is to extract glutathione from thiols. And so we can actually do the experiment in the presence of Glutathionylation and completely reverse the effect of glutathionylation on the unfolded protein. But in vivo, these measurements have not been done. So, I don't know the answer to that. But I think that intuitively, we kind of know the timescales that it takes to stiffen up and then, to sort of soften your muscles. I mean, we're talking about hours probably in vivo. It'd be very interesting to find out, actually.
It's a beautiful picture that this titin performs work when it gets collapsed.
When it falls.
When it falls, yeah. But if it falls very rapidly, then most of this work immediately dissipates on the spot. It cannot perform work on anything else. So from this point of view, when a sportsman, a baseball pitcher performs this motion, it's very rapid motion, it probably becomes very inefficient when it's very rapid.
Two things. Folding shortens under force. So it does work. Mechanical work. How many of these events happen as a function of time is really what you're asking, because it's not going to be one folding event, it's going to be a large number of folding events in the contracting muscle. And so we're talking about distributed timescales over a large number of titin molecules, 10 to the 7, 10 to the 8 titin molecules, first of all. The second is that, as I explained before, stretching creates a pool of unfolded modules from which you draw. So you're not asking the probability of one folding to take place, but 1 out of maybe 50 or so. And that can happen on very fast time scales. The last point I want to make is that, and I didn't explain this, but maybe it's self-explanatory. We have some potential and mean force models for the folding and the unfolding of a tandem modular protein. And so what you can see here on the right is a simple consequence of the model. So this is the folded protein. When you change the length, how much force it takes to get that much length change. And total amount of energy that you can store is the area under this curve.
So this is like Hookean deformation without...
Well, it's not Hookean, it's really the worm-like chain. But it's the same idea, right?
Yeah.
But in contrast to a spring, a Hookean spring, right, you have that this is a nonlinear force-length relationship, and second you have unfolding, okay? So
but it's slightly more.
You can go up very quickly here, and then you unfold.
Yeah, this is real.
And you go here, right? So if you do this very fast, some of the energy is going to be burned as heat, because you're going to get hysteresis on the return. This is what you were alluding to, right? So if you do it very fast, you're going to increase the amount of heat dissipation, but you're still going to have an enormous increase in the amount of elastic energy that you store and that you recover from the fold. If you go up more slowly, you can go up and down this green curve here, which represents the equilibrium folding and unfolding. And if you do that, there's no heat dissipation. But you're going to throw a ball, very likely you're going to go as high as you can here quickly and fold, and then maybe you're going to have recovery, which is going to be on a different time scale. And that's going to create both energy dissipation as heat, but still you're going to increase 20, 30 times the amount of elastic energy that you recover by having the folding reaction. So, I mean, evidently there is a price you pay for the speed, and that price you pay is heat exchange. And that is something that we all experience.
Any further questions? If you were to design an artificial muscle, if you went down that route, are there structural bits of information that you've acquired along the last couple of decades that you would put in an artificial muscle? Do you want to have lots of beta sandwiches instead of alpha helices?
I'm a humble observer of God's work. I have no clue.
Are helical proteins better than beta, anti-parallel beta sheets?
So let me say
of
something. We have enough genomes today, and we were talking about this earlier, that people have sequenced titin in lots of different species.
Mm-hmm.
And you can actually reconstruct the evolution of the titin sequence over hundreds of millions of years. I mean, all the way up to the Cambrian explosion 500 million years ago. What one of these evolutionary biologists told me is that the rate of mutations in titin is the largest measured for any other protein that has evolved over time. Mm-hmm. So very clearly there's something about this protein that's very crucial for the environment in which the organism lives that requires this adjustment. Okay? And so, evidently we have optimized this for the kinds of activities that we do, the gravitational field on Earth, and the mountains, the swimming, all of the things that are essential for surviving. If you want to think about something profound, imagine that now we send people to Mars. Their titans are going to be totally off. And maybe there's no roadmap in the genome to let them adapt in a way that's meaningful and that preserves health. So I mean, I don't know if there are any NASA people here, but I can tell you that don't mess with Titan, because Titan has taken 500 million years to get to this point that allows us to go up the stairs. You put something on a different gravitational field, how much energy can be stored, the kinds of motions that you take advantage of for optimizing your energy consumption, all of this is going to be ruined.
So Deco has a last burning question, and then we'll take it upstairs.
If you send the bear to Mars, and the bear hibernates,
And does
what isoform would the bear put in his heart? That should keep you awake at night.
So what happens?
I wish we could.
Titan is optimized, you're saying, for Earth's gravity, but that means it should be optimized for different animals that are, say, different size and stuff like this. So,
Very likely. We don't know this is very likely to be true. I just don't know the answer to it.
Okay, so
It has
it's
not
something
been
that
studied.
obviously one could figure out.
We can, but it's going to take another 50 years of NIH funding.
So if you follow up on Titan, if you look then like at whales versus cows, right, you should see a big change in Titan relative to other proteins, right?
Very likely the way the cow uses Titan and the way the whale uses Titan is radically different.
But, so that would be a place you'd expect to see that enhanced mutation,
It would
right?
be kind of hard to justify the research, but I agree with you.
Well, just sequencing, right? It probably exists.
Well, I think that what actually you want to do is take the animal, put the animal and the different kinds of mechanical activities and see how the animal is controlling the Titan isoforms, because they're not permanent. As I just gave the example, okay, let me give you a more radical example. I'm collaborating with a couple of surgeons at Columbia that are interested in the mechanobiology of birth. The uterus is tiny, but it becomes the largest muscle in the human body during pregnancy. I mean, think of the octomom, okay, with octuplets. All right, sorry, John.
Yeah.
So evidently, this cannot be just a rubber band that you stretch. There are massive readjustments in the Titan isoforms in the growing muscle that has only one function. When the time comes, it has to contract, and push the babies out, right? And so this tissue has to become from very rigid and small to insanely elastic and huge, right? Actin and myosin are always the same, no change. The main difference is Titan. We've been running gels, finding out that Titan is actually switching. So there are all these adaptations. So when you think of Titan, don't think one Titan per muscle. Think of something that's very dynamic. So I love to have, for example, human samples taken from an athlete that, let's say, runs marathons, but before and after the training, right? It's a little problematic to do this, but eventually we're going to end up doing these things, right? And then we're really going to know. I don't know how anybody could persuade graduate students to take sample from hibernating bears, okay? But
Hibernating squirrels, right?
well, I mean, the bear was a bit more impactful, I think, than the squirrel. But I mean, it's amazing the shift in Titan isoforms, in the heart. The same happens during a heart attack when you have an infarct. The heart becomes dilated, and the speculation is that also there is a switch in the isoform that causes the dilation of the tissue, and that's problematic because then you have a hard time contracting and expelling blood. There are all these sort of stories that probably converge on one question. Titan has been regulated to adjust elasticity for the particular form of motion that you need at that time. It's not a static thing. I love to take samples from astronauts right? When they come back to Earth, there's a paper actually from a Russian group, that took samples from astronauts, but the conclusions, the techniques and the approach was, the idea is exactly the same. See if titin is still there, and if the isoforms had switched. The results were not clear, but that experiment needs to be done eventually.
Okay, so I warned you that Julio was going to tell us something radical and something new, and so I think we satisfied this goal wonderfully, and let's thank Julio again.